Hello, everyone. It is a great honor to share our latest research findings with you. The gut microbiome is closely linked to the host's endocrine system, and together they influence host reproduction, stress response, and physiological health. Hormone levels such as estrogen, testosterone, and cortisol serve as important indicators for assessing reproductive status and stress adaptation. Microorganisms can help regulate host sex hormone homeostasis through mechanisms such as the degradation or reactivation of steroid hormones. Research on the dynamic associations between microbiome and hormones across different physiological stages and sexes remains very limited. The highlight of this study lies in the systematic analysis of gut microbiome and variation characteristics across different physiological stages and between sexes, as well as the exploration of potential correlations between them, by collecting fecal samples from giant pandas at different physiological stages and integrating sex determination, hormone assays, and metagenomic sequencing. The study performed individual identification on 62 high-quality samples. By combining sex determination, bite mark measurements, and microsatellite data, a total of 32 giant pandas were identified, including 16 males, 12 females, and 4 individuals of undetermined sex. We compared the gut microbiome of giant pandas across different months and between genders. The results showed that gut microbial alpha diversity fluctuated by month, with higher levels observed in June and July. PCOA and distance-based analyses further revealed significant differences in microbial composition across months. When comparing males and females, we observed some differences in both the Shannon index and beta diversity across months. However, none of these differences reached statistical significance. Species composition analysis revealed clear monthly fluctuations in the gut microbiome of the entire population, as well as in both males and females giant pandas. At the genus level, in March and April, the gut microbiome of both sexes was dominated by Pseudomonas and Clostridium sensu stricto 1. In July, males were dominated by Acinetobacter and Lactococcus, while females were respectively dominated by Flavobacterium and Lactococcus. In addition, linear discriminate analysis and statistical testing revealed significant differences in certain microbial strains, between males and females, and across different months. Hormone levels in male and female giant pandas also showed dynamic changes across different months. In females, estrogen peaked in March. Progesterone remained elevated from March to June, and testosterone also exhibited seasonal fluctuations. Monthly variations in estrogen and progesterone were also noted in males, while testosterone levels were higher in March and July. Notably, in both sexes, cortisol levels were higher during the bamboo leaf and stem feeding phases compared to the bamboo shoot feeding phase. This may reflect adaptive hormonal regulation in response to dietary nutritional stress or other survival pressures. In addition, our study revealed significant microbiome endocrine coupling in giant pandas. We used network and Spearman correlation analyses to assess this coupling and found widespread significant associations between microbial taxa and hormones. Microbial alpha diversity was positively correlated with progesterone and estrogen, but negatively correlated with cortisol and testosterone. Notably, the genus Lifsonia, which showed a positive correlation with estrogen and progesterone, also exhibited a corresponding increased abundance during March and April in the estrus period, when the levels of these two hormones were higher. Furthermore, analyses at both the sample and individual levels showed that this association was significant in males but not in females. Accordingly, males exhibited greater variability than females in both their hormonal profiles and gut microbiome composition. In summary, this study yielded 3 main findings. First, the gut microbiome composition of giant pandas varied across seasons and genders. Second, hormone levels in giant pandas showed adaptive regulation in response to reproductive activity and metabolic status. Third, the microbiome hormonation was detected in giant pandas, which is significant in males, but not in females.